So my name is Rolando Ceddia, and I'm from York University. I'm going to chair this session. So we have 4 presenters, and we are going to start with so the order of presentation will be Dr. Kristen Stanford, who is with us already, and followed by Dr. Lawrence Kazak, and then Dr. David Wright, and I will finalize, I'm the 4th presenter. So each person will have approximately 25 minutes, and let's hope we have 5 minutes for questions, so the audience can interact with the presenters. So I'm going to start and introduce Dr. Stanford. So she did a master's in kinesiology in Indiana University, and then she did a PhD in biomedical sciences at the University of California, so you moved west, yeah. And then a postdoc back in East again at Joslin Diabetes Center, Harvard Medical School, under the supervision of Dr. Laurie Goodyear. So she's currently an associate professor at the Ohio State University, Wexner Medical Center in Columbus, Ohio. So it's a great pleasure to have you here, Kristen. So we look forward to your talk. So please take it away.
So thank you all for being here. I want to thank the organizers. This is my first IBEC, so I'm very excited to be here and talk about some new work from our lab looking at exercise-induced adaptations to brown adipose tissue. Which one is the advancer? That's this big green button. Got it. So I really need to remind this crowd, but there are several beneficial effects of exercise on health, and I won't go through this list, but part of why we've been interested in this is because exercise plays these really important roles to prevent diabetes, and this includes improving insulin sensitivity, glucose tolerance, mitochondrial function, increased muscle glucose uptake, and improved pancreatic function. And so we know that exercise has beneficial effects on the skeletal muscle, and in more recent years, people have focused on several other tissues that are affected by exercise and can improve whole body metabolic health. And we've become really interested in the effects of exercise on brown adipose tissue. Kind of the central question that my lab focuses on, or one of them, is do exercise-induced adaptations to brown adipose tissue mediate the beneficial effects of exercise on metabolism and cardiovascular health? And again, with this general idea, if these exercise-induced adaptations to brown adipose tissue can really facilitate improvements in several other tissues. So when we talk about adipose tissue in humans, adipose tissue can be broadly subdivided into brown and white adipose tissue. And so brown adipose tissue in an adult human is found in the cervical and supraclavicular and paravertebral regions. In infants, it's in this intrascapular depot, which is also where it's found in rodents. And it's really involved in energy expenditure. White adipose tissue is found in the subcutaneous depots in humans, as well as in the intra-abdominal region, and can also be found in other sites, including the retroorbital areas, periarticular regions, bone marrow, intramuscular, and pericardial. I wanted to give a little overview on the differences between brown and white adipose tissue. So white adipose tissue gets its name because it's white in color. It's vascularized by an arteriole. A white adipocyte is 50 to 105 microns in size. It has a unilocular fat droplet, which you can see both by this cartoon and this H&E stain. It has few mitochondria, peripheral nuclei. Its unique marker is leptin, and it plays a primary role in energy storage. When we talk about brown adipose tissue gets its name because it's brown in color. It has multiple capillaries. The size of a brown adipocyte is a little bit smaller than a white adipocyte. It's 38 to 45 microns in size. It has multilocular fat droplets, abundant mitochondria, a central nuclei. Its unique marker is uncoupling protein 1, and its primary function is in energy expenditure and thermogenesis. And so part of why we become interested in brown adipose tissue and obesity is because that is this highly energetic organ that plays an essential role in energy balance and thermogenesis. And these 3 seminal papers published in 2009 showed that brown adipose tissue is inversely correlated with fat mass and BMI in humans. And what they did is, this is the the same subject at room temperature or after 4 hours of cold exposure. And the 4 hours of cold exposure were wearing shorts and a t-shirt at 65 degrees Fahrenheit with ice being rubbed up and down their legs. And when they did this and they did the PET scan following the cold, they could now detect these symmetrical lesions of brown adipose tissue in the supraclavicular and perivertebral regions. They showed that this brown adipose tissue was correlated to energy expenditure. And they also showed that brown adipose tissue was correlated with age. So subjects who were over 65 had less detectable BAT than those under 50. It was correlated to BMI. Subjects with a BMI of more than 27.8 had less detectable BAT than those with a BMI of less than 23. And also correlated to fasting glucose. So subjects with a fasting glucose of over 103 had less detectable BAT than those with a fasting glucose of less than 93. So this led to the idea that increasing the amount of brown adipose tissue could be a useful tool to combat obesity. And so when I was in Lori Goodyear's lab, one of the things that we wanted to look at was if we could increase brown adipose tissue and improve glucose tolerance. And so what we did in this cohort of mice is we took 0.1 gram of brown adipose tissue and transplanted it into the visceral cavity of a recipient mouse. So this is essentially a doubling of their brown adipose tissue. This data, our glucose tolerance, is area under the curve. These black bars are the sham mice. The red had a bead as a space control. The blue bars had received 0.1 gram of visceral white adipose tissue, and the white bars received the brown adipose tissue. We looked at 2, 4, and 6 weeks post-transplantation. We really didn't see a difference. But when we looked at 8, 10, and 12 weeks post-transplantation, we saw that the mice that received brown adipose tissue had a significant improvement in glucose tolerance. To show this slightly differently, what you can see is that these sham mice, the mice that received the bead, or the mice that received the white adipose tissue, had this age-induced worsening of glucose tolerance, which is what you typically see in black 6 mice. But the mice that received brown adipose tissue were protected from this. So when we start talking about brown adipose tissue and exercise, the effects of exercise on brown adipose tissue have really been conflicting. Some studies indicate that exercise increases BAT activity in humans. Some suggest that it decreases BAT activity in humans. And some studies have shown that athletes had a decrease in cold-stimulated brown adipose tissue activity compared to lean controls. And so this is a representative picture from this paper that came out in 2015. And so these subjects are male, age, and BMI matched. This is a self-reported trained subject compared to an untrained subject after 2 hours of cold exposure. And you can see that the untrained subject has more detectable brown adipose tissue than the trained subject. So what we decided to do was really ask a really broad question, which are what are the effects of exercise on brown adipose tissue? And I'm just going to summarize this work in one slide. What we saw in rodents was that after anywhere between 2 and 3 weeks of exercise, it decreased the cell size of brown adipose tissue, decreased its lipid content, but it didn't alter mitochondrial markers. Exercise did not change thermogenic markers in brown adipose tissue. It decreased basal mitochondrial activity, and it decreased basal glucose uptake. And so we were a little bit puzzled by these data. I'm an exercise physiologist by training, so I don't like for exercise to not do anything good or have a kind of a blunted effect. So we started trying to think about what we were looking at and what could be different, and realized that we weren't taking into consideration the fact that adipose tissue is an endocrine organ. And so we know that adipose tissue releases these adipokines, and they can be broadly categorized into pro-hyperglycemic or anti-hyperglycemic. And so we decided to ask if exercise could alter the endocrine role of brown adipose tissue. And to do this, we decided to look at signaling lipids. And so this is this emerging class of hormones. And in 2008, palmitoleic was identified to be released from adipose tissue and improve insulin action. Studies in 2013 identified these phosphatidylcholines to be released from adipose tissue and increase muscle fatty acid uptake. Studies in 2014, Barbara Kahn's group identified these passes to be released from adipose tissue and improve glucose tolerance. And work by our collaborators in 2017 identified 12-13-dihome to be released from brown adipose tissue to improve fatty acid uptake. So we decided to ask if exercise could alter signaling lipids that could improve metabolic health. This was done in collaboration with Paul Cohn's group. He's at Advent Health, and this has really just been a phenomenal collaboration. What Paul was able to do is to take healthy, can I go back? Maybe, yeah. To take healthy male subjects, a very broad age range, 24 to 90. We took a pre-exercise blood draw, had them exercise on a cycloergometer for 40 minutes. Took a blood draw immediately post-exercise, and then again 3 hours after exercise. We sent these samples to Berg Health with Michael Kiebisch, and he ran shotgun lipidomics on the samples. We detected 88 lipids with annotated signaling properties. When we looked at these samples by volcano plot, what we saw was this one lipid was really increased with exercise. And it turned out that this lipid was 12,13-dihome. So what is 12,13-dihome? Well, previous studies had shown it was a bradykine. It's released from brown adipose tissue. It's a linoleic acid metabolite. It's acted on by the CYP450, resulting in 12,13-epome, which is then acted on by these soluble epoxide hydrolases, resulting in 12,13-dihome. It's released from brown adipose tissue in response to acute and chronic cold chronic cold in mice and humans, and it increases fatty acid uptake into brown adipose tissue. When we looked at these data, each subject individually, we saw that 12,13-dihome was increased in every subject after exercise. When we quantified it, we can see that it significantly increased immediately post-exercise, but trending back down 3 hours post-exercise. To confirm and extend these findings, we collaborated with Lori Goodyear and Jan Willem Middelbeek at the Joslin Diabetes Center. And what they were able to do was to take healthy male and female subjects, aged 27 to 31. They took a pre-exercise blood draw. These subjects were then run on a treadmill, so a different exercise modality. They took blood 15 minutes into the exercise, immediately post-exercise, and then 1 hour post-exercise. And what we saw, so the female subjects are in pink, the male subjects are in blue. Each subject, with the exception of these two, had an increase in 12,13-dihome immediately post-exercise, and then again back down to baseline 1 hour post-exercise. So now we're seeing that this effect is an increase in 12,13-dihome in both male and female subjects. We then wanted to ask if this increase in 12,13-dihome correlated to functional metabolic changes in humans. And so this data is both the Florida cohort and the cohort from Boston. What we saw was that BMI was negatively correlated with 12,13-dihome. Total fat mass negatively correlated with 12,13-dihome. Triglycerides were negatively correlated with 12,13-dihome. And because we were really interested in exercise, we also saw that we looked at VO2 peak and saw that VO2 peak was positively correlated with 12,13-dihome. So these data showed us that this increase in 12,13 is correlated to functional metabolic changes and improved cardiorespiratory fitness in male and female human subjects. So we then wanted to ask what tissue is responsible for this increase in 12,13-dihome. So to do this, we went back to our mouse model, and the first thing we did was measure if 12,13-dihome was increased in mice after an acute bout of exercise. So this is serum immediately post-exercise, and we see a significant increase in 12,13-dihome. And then we asked if this exercise-induced increase was from brown adipose tissue. And this is because our colleagues had seen that cold-stimulated 12,13-dihome to be released from brown adipose tissue in mice. So to do this, we took mice, we surgically removed their brown adipose tissue, we waited 8 weeks, and then subjected them to an acute bout of exercise. And we did this, you can see these are the sham-operated mice, the sedentary mice, and then an increase in 12,13-dihome immediately after an acute bout of exercise. And these are the sedentary mice that do not have brown adipose tissue, and then after an acute bout of exercise. And so when we did this, we saw that there was no exercise-induced increase in 12,13-dihome in the mice that did not have brown adipose tissue. So we then wanted to ask what the physiological function of an exercise-induced increase in 12,13-dihome could be. And we hypothesized that anything coming from brown adipose tissue in response to exercise would likely affect the skeletal muscle and the heart. And so first we wanted to ask if 12,13-dihome could increase fatty acid uptake and oxidation in skeletal muscle. To do this, we took actocreme mice, crossed to luciferase mice. These mice would then express luciferase only in their skeletal muscle. Andreas Stahl's group developed this assay, which is a free fatty acid conjugated to a luciferin. So when we inject these into mice, now we can detect fatty acid uptake specifically in our tissue of interest. And so we hypothesized that when we injected free fatty acid luciferin in a vehicle and free fatty acid luciferin and 12,13-dihome, that we would see an increase in fatty acid uptake in the mice that receive the 12,13-dihome. So this mouse over here on the left received the 12,13-dihome and the free fatty acid luciferin, and this mouse received the vehicle. Play for me. And what you can see is a significant increase in fatty acid uptake in the mice that receive the 12,13-dihome. When we quantified it, you can again see the increase in fatty acid uptake. It's pretty rapid. This is over only 12 minutes, and we see a really significant difference in the first 4 minutes after injection. We also wanted to look at cardiac function, and around this time, a couple other studies had come out that had indicated the brown adipose tissue was protective in a model of cardiac function or in cardiovascular disease. And so we injected 12,13-dihome into the carotid artery of mice and measured systolic and diastolic function, and saw a significant increase in or improvement in systolic and diastolic function in these mice. And these are chow-fed 12-week-old mice. So these data show us that 12-13-dihome is increased in circulation in humans after exercise. Brown adipose tissue is the main source of 12-13-dihome. 12-13-dihome increases fatty acid uptake and oxidation in skeletal muscle, and it increases systolic and diastolic function. So because we saw this effect of exercise to increase 12-13-dihome, and we saw that in the absence of brown adipose tissue, we didn't see this exercise-induced increase in 12-13-dihome, we wanted to ask if there was a similar effect of exercise and increasing brown adipose tissue on signaling lipids. And this project was really driven by Kelsey Pinkard, who was a graduate student in my lab. She's now a postdoc in John Stafford's lab, and Vikram Shettigar, who's a postdoc in Mark Ziolo's lab at Ohio State. And so what we did is we took brown adipose tissue from donor mice and transplanted it into the visceral cavity of recipient mice. We also added a group of sham mice. To show you how we did the transplant, we take the brown adipose tissue and place it right on top of the perigonadal fat pads. And we've previously shown that sympathetic innervation in these mice is confirmed 12 weeks post-transplantation. We then added an exercise paradigm. So we had 3 groups of mice. We had our sham sedentary mice, we had our plus bat sedentary mice, and then we had mice that underwent the sham surgery. We waited 4 weeks, and then they underwent an intense interval exercise training program for 8 weeks. When we took their blood and we quantified their lipids, we saw that there was one signaling lipid that was really increased in the mice that received brown adipose tissue. And we also saw that there was one lipid that was really increased in the mice that exercised. And in both cases, that lipid was 12-13-dihome. So 12-13-dihome was the most significantly upregulated lipokine in response to both increasing brown adipose tissue and exercise. All right, so the data that I've shown you so far has shown that we've seen an improvement when we injected 12-13-dihome acutely. We saw a really rapid improvement in muscle fatty acid uptake. We saw a very acute effect on cardiac function. We wanted to ask if there was an effect of a sustained increase in 12-13-dihome to affect both metabolic and cardiac function. And what's tricky about these oxylipins is that they're very quickly metabolized. So we've done a couple studies where we've injected, our lab and Yuval Singh's lab have injected mice over a period of 16 weeks, daily injections with these lipids, and you can't detect them in the bloodstream because they're metabolized within 4 to 7 minutes upon injection. So we wanted a way where we could actually result in this sustained upregulation of the lipid. To do this, we collaborated with Daniel Geigo-Perez's lab. And Daniel's a bioengineer, and when he first presented at OSU, I thought that he did magical things, and I basically told him that. So what he's developed is this technique called tissue nanotransfection. So he can take your plasmid of interest and transfect it in the skin. And what we had hypothesized would happen is that if we took these plasmids of EPHX1 and 2, which are the enzymes that make 12-13-dihome, that we would see an upregulation of 12-13-dihome in circulation. And when we first started talking about this, he'd only tested this at a really molecular level, so we didn't know if it would work. But what we did is we put mice on a high-fat diet for 6 weeks. We then did weekly transfections of TNT with either EPHX1 and 2, which again are these 2 enzymes that convert 12-13-epome to 12-13-dihome, or of UCP1, which is a marker of brown adipose tissue. We did it once a week for 6 weeks. We then waited 2 weeks and then performed several in vivo analyses. So first we measured 12-13-dihome in the blood. And again, this is 2 weeks after our last treatment with TNT. And what we see is that when we look at the mice that received TNT with EPHX1 and 2, we see a significant increase in 12-13-dihome. What was really important is that this is about a 2-fold increase in 12-13, which is similar to what we see with exercise and what our colleagues have seen with cold exposure. There was no increase in 12-13 in the mice that received the TNT with UCP1. When we looked at body weight gain, we saw that TNT with EPHX1 and 2 attenuated body weight gain. These mice are still on a high-fat diet. This was improved compared to both the UCP1 mice, the mice that received TNT with UCP1, as well as our sham mice. When we looked at what was affecting this difference in body weight, we saw that TNT with EPHX1 and 2 decreased fat mass in these mice. They had a slightly elevated lean mass. When we looked at cardiac function, this is by pressure volume loops, we saw that sustained increase of 12-13-dihome prevented the high-fat diet-induced decline in cardiac function. So these are the mice that received TNT with EPHX1 and 2 compared to both the sham and UCP1 treated mice. So these data showed us that 12-13-dihome is increased with increasing BAD and exercise. This sustained increase of 12-13-dihome attenuates body weight gain and preserves cardiac function. And we've been really interested in these data, but these are all in mice. And so we wanted to see if there was a translational component to this. And I'd previously shown that we saw this increase in 12-13-dihome with exercise, but we hadn't looked in any human disease states. So we wanted to ask if 12-13-dihome was altered in patients who have either type 2 diabetes or cardiovascular disease. And so again, we collaborated with Paul Cohen's lab, and what Paul was able to do was to take human samples from these 3 clinical trials. So we had patients who were healthy, patients who were obese, patients who had cardiovascular disease, or patients who had type 2 diabetes. We had both male and female patients, an average age of 57. The obese population was defined as having a BMI of greater than 30 with no type 2 diabetes or cardiovascular disease. And I will say that the cardiovascular disease was very broadly categorized, so basically anyone who had any type of cardiovascular dysfunction. And so when we looked at these patients by BMI, what we can see is that the patients who are obese, have cardiovascular disease, or have type 2 diabetes all have a very similar BMI, elevated compared to the healthy subjects. When we looked at total lipid abundance, we see that the patients who are obese or had type 2 diabetes have slightly elevated plasma lipid abundance. And then this, to me, was the most exciting part of this whole study. So when we looked at this PCA plot, what you can see is that there's a distinct effect of the patients who have cardiovascular disease versus patients who have type 2 diabetes. So this is signaling lipid data, and you can see that they cluster very differently. Our healthy subjects are right in the middle in these dark blue dots, and the patients who are obese kind of fall some with the patients who have type 2 diabetes and some who have cardiovascular disease. And so we're not really sure what the meaning of that is. Kind of my thought is that these could potentially be used as biomarkers if we can figure out if these patients who are over here who are obese are more likely to develop cardiovascular disease in the future, but that's kind of where this work is going. Okay, back to our heat map. So this was a 2-way hierarchical clustering in these signaling lipids. And what you can see is a distinct effect of patients who have type 2 diabetes, who are healthy, patients who have cardiovascular disease, or who are obese. And these clusters divided into about 7 different clusters. I'm just going to pull one of them out for right now. What you can see is that this is cluster 1. So this was elevated in our healthy subjects. When we look at the lipids that were in this section, what you can see is one of interest to us really popped out in 12-13-dihome. So 12-13-dihome is up in the healthy patients compared to all other groups. And in fact, when we quantified this by volcano plot, what you can see is that there's only one lipid that is decreased in each disease state, and that lipid is 12-13-dihome. So it's decreased in patients who are obese compared to healthy, in patients who have cardiovascular disease compared to healthy patients, and in patients who have type 2 diabetes compared to healthy subjects. We also looked at BMI and blood glucose, and again, this is now the 3rd patient cohort we've looked in for this, and again, we see that 12-13-dihome is negatively correlated with BMI and also with fasting glucose. So these data showed us that obesity, type 2 diabetes, and cardiovascular disease result in distinct lipokine profiles in patients. 12-13-dihome is decreased in patients who are obese, have type 2 diabetes, or have heart disease. 12-13-dihome is negatively correlated with BMI and fasting glucose. And our overall conclusions are that brown adipose tissue is an important tissue to regulate whole body metabolic homeostasis and cardiac function. 12-13-dihome is a bat-release lipokine that we see regulated by exercise that plays this important role in metabolism and cardiac function. And obesity, type 2 diabetes, and cardiovascular disease result in distinct lipid profiles in human patients. And our hope is that this could eventually be used as a diagnostic tool or therapeutic target. I just want to conclude by thanking the people who did the work. This is our most recent internal research day, but Lisa Baer, Diego Hernandez Savadra, Kelsey Pinkert, and Catherine Wright really drove this project. I also want to mention Shinsuke Naringi, who's here, and he has a wonderful poster number 38, as well as Pablo Vidal, who is also here and has poster number 4. I'd like to thank our collaborators at Advent Health, especially Paul Cohen. It's really just been fantastic what we've been able to do with his help and assistance. At Ohio State, Mark Ziolo kind of drug us into the cardiovascular aspect of things. Daniel Gallego-Perez has been this bioengineer who's done these really fantastical things. Doug Lewandowski, Lauren Wald, and Ty Saldana have helped us, again, with some of this cardiovascular aspect, and then our collaborators at Berg Health who have run all of our lipidomics and our funding sources. And happy to take any questions.
Excellent, Christine. Great timing.
Please, if you have questions, the microphone is open.
Me or?
Yeah, bringing
Okay,
it.
yeah. Thank you very much for a nice talk. Very interesting. How do you think that exercise is leading to the synthesis of 12-13 diahome, and what goes wrong in disease that 12-13 diahome goes down?
Yeah, it's a great question, and I don't have a great answer for it. I think it might have to do with how the tissue is innervated, and if we see an increase in sympathetic activity that could stimulate that. But we're not really sure at this point. But that's kind of our working hypothesis.
And people on beta blockers and things like that, which sort of inhibits all that, do they have changed concentrations or?
Yeah, that's a great question. We've not looked at that, so we're trying to get some human samples right now where we can actually ask that question.
Okay, thank you.
Thanks.
Hi, thanks for the talk. So you showed really nicely that it's bat releasing the 12-13 dihome in mice. And is there a way you can show that in humans?
Yeah.
Or could it be coming from somewhere else?
Yeah, so that's a great question. So what we've seen in mice, so we've looked in several tissues in mice and shown that with either exercise or cold exposure, the only tissue that increases the actual lipid production as well as increases the enzymes that produce the lipid is brown adipose tissue. We can't really do that in humans, although we're trying. So we're trying to look in patients who are in different disease states who might undergo a surgery to see if they have different expression of EPHX1 or 2 in their brown adipose tissue, but we're still kind of at the baby steps into that aspect.
That sounds great, thanks.
Anybody else? So thank you very much, Christine. Okay, before you start, Lawrence, let me introduce you. Okay, our next speaker is Dr. Lawrence Kazak. I first met Lawrence when he was still a master's student at York. It's some time ago. I'm not going to say too long because he's still quite young. And then he went on to do a PhD in biological sciences at the University of Cambridge. Then he moved to a postdoc at Dana-Farber Cancer Institute at Harvard Medical School under the supervision of Dr. Bruce Spiegelman. Then he currently is an assistant professor at the Department of Biochemistry, Rosalind and Maurice Goodman Cancer Institute at McGill University in Montreal. So please, Lawrence, thanks very much for accepting this, and we look forward to your
Yeah, I had nothing better to do, so thanks. So, yeah. Thanks, Rolando. So my talk today is not going to be about exercise at all, but it's going to be about new players and adaptive thermogenesis. So brown adipocytes are densely packed with mitochondria, where the oxidation of substrates such as fatty acids, glucose, succinate, branched-chain amino acids. Electrons are stripped from these macronutrients, and protons are pumped into the intermembrane space to generate a proton motive force. And these reactions are all stoichiometrically fixed, so in order for a cell to generate heat, there are essentially two ways that can occur. Either substrate oxidation could be uncoupled from ATP synthesis through proton re-entry into the matrix through uncoupling protein 1. But also ATP turnover can be accelerated. If that's not coupled to the production of work, that would also generate heat. We love UCP1. We have projects on UCP1. We know it's important, but I'm going to focus on work that we're trying to understand how ATP sinks are used in adipocyte thermogenesis, because a lot less is known. We think it's important. So I'm going to start with the model of ATP turnover, which we call the futile creatine cycle. And hopefully by the end of the talk, I'll convince you that we have some pretty good evidence that this model is probably correct. So here we have electrons flowing to oxygen, protons are pumped to the intermembrane space. 3.7 protons are required to generate 1 molecule of ATP, and so if you divide 10 protons by 3.7, you get this number, which is a P to O ratio. So we use the P to O ratio to calculate ADP liberation by monitoring oxygen consumption. So when you stimulate respiration with ADP in mitochondria, this stimulates ATP. ATP gets exported out. And then a creatine kinase, which we know in at least murine brown adipose tissue is CKB, the reactants and products of the CK reaction are all in a one-to-one stoichiometry. But when we take thermogenic mitochondria and we give a quantity of creatine, and we measure how that affects oxygen consumption, we get an excess of oxygen consumption, a molar excess of ADP liberation. So about 6 years ago now, we developed this model where we thought, okay, if creatine is stimulating a molar excess of ADP liberation, maybe there's a regeneration step, maybe through multiple steps, maybe one enzymatic step. And Bruce's lab has just identified last year tissue nonspecific alkaline phosphatase as a hydrolysis step for phosphocreatine. It regenerates creatine, and every cycle here liberates ADP, which stimulates respiration, right? Uses electrons for macronutrients and thus heat production. So in my lab, we focused on the creatine kinase angle, so I'm just going to briefly go over some published work. So here we use quantitative proteomics to ask which creatine kinase isoform is most important in brown adipocytes, and it was pretty clear from this data that CKB is the only isoform that's expressed. CKM, CKMT1, CKMT2 were not detected at all. So we made knockout cell lines, so we have 2 independent clones that express CKB, 2 clones that are completely devoid of CKB. There's no effect on adipocyte differentiation. And then we did the critical experiment where we traced deuterated creatine into deuterated phosphocreatine in brown adipocytes. And you could see in black and in gray, these are the 2 clones that express CKB. You can generate an M+3 isotopolog of phosphocreatine. And in the 2 clones that don't have CKB, you don't get any phosphocreatine. And importantly, when you trace creatine in, creatine is actually accumulated. So this defect in phosphocreatine abundance is not due to a defect in creatine influx. Creatine is getting in, it just can't be converted to phosphocreatine. So we think this is pretty good data, evidence to suggest that CKB is probably the primary creatine kinase in murine brown adipose tissue. Okay, that posed a problem though, because the futile cycle is supposed to happen in mitochondria, and CKB is not annotated as a mitochondrial protein. But based on our quantitative proteomics data, it was the only isoenzyme that we found, so we hypothesized that maybe it's dually localized. I'm going to show you some data about that. So here we used immunofluorescence, where we express CKB flag, TOM20, which marks mitochondria, and you could see co-staining of CKB in TOM20. A lot of it is not mitochondrial, but a substantial portion is, and those are the white dots. We also used a biochemical approach where we isolate intact mitochondria, we treat with trypsin. And you could see when you infect brown adipocytes with CKB flag, in the presence of trypsin, it's completely resistant to proteolysis, as is TIM23 and TFAM. These are mitochondrial markers. But outer mitochondrial membrane proteins like TOM20 or ER proteins like calnexin, peroxisomal proteins, PMP70, those are all degraded. We went a little bit further. We wanted to understand how CKB is imported into mitochondria. Because it lacks a classical N-terminal mitochondrial targeting signal. But what we found, and this was kind of inspired by Johannes Hermann's work, this is not our idea, he identified these internal mitochondrial targeting signals. And we could see that CKB has a very large signal around amino acid 120. Now, there are known to be arginine residues that are important for these. And clear enough, there are 4 arginine residues in this region. And we predicted if we mutated these to glutamic acid, we should lose this signal. So now we test experimentally whether this is the case. So here I'm showing you flag Western blot. What's important to follow is just these 4 lanes here. These are all mitochondria, protease protected from CKB knockout brown adipocytes, and then rescue experiments. So we add GFP, wild-type CKB, the delta MTS mutant, so this is the import dead mutant that we predicted, and then a catalytic dead CKB. And you could see that wild-type CKB and the catalytic dead are imported, they're protected from proteolysis, but the delta mutant is obviously degraded. And then we can look at CK activity, right? So we can take control mitochondria, lots of creatine kinase activity, the knockouts with GFP, it's gone completely. We can restore CK activity in the wild type. The delta MTS, there's no activity because it's not imported, and the catalytic dead, it's present, but it's catalytic dead. Okay, at this point you might say, well, you made a bunch of mutations and maybe your protein is unstable. That's not the case, because you could take cytosolic extracts from the same samples and you could see the protein expression of all 3 variants is equal. And more importantly, when we do the rescue experiments, wild type and the delta MTS have identical CK activity. Okay, so mutation of the internal MTS is responsible for import CKB. Okay, at the same time, Yitzi's son in Bruce's lab identified tissue nonspecific alkaline phosphatase to also be localized in mitochondria of brown adipocytes, and they showed by NMR that TNAP had the predicted activity of a phosphocreatine hydrolase. Okay, I'm just going to show you one slide. Anyone that's interested in actually doing these experiments properly, please feel free to email me at any time, and I'm happy to talk. So this is what a typical experiment might look like with butyl creatine cycling. So we use a Clark-type electrode, we add ADP, and then we just monitor oxygen consumption from the state 3 to state 4 transition here. And importantly, what we do is we just calculate simply the creatine-dependent change in oxygen consumption. We know how much creatine we added. We can predict based on the P to O ratio how much ADP was liberated, and then we calculate a stoichiometry. And when we do this, it doesn't really matter what cells. If we use immortalized brown adipocytes or primary, UCP1 knockout, they all hover around 20, which means that creatine seems to be, at least under these conditions in this system, cycles 20 times. That's what a stoichiometry of 20 means. And importantly, if you deplete CKB, either genetic knockouts or knockdowns, we don't see any superstoichiometric effect. We wanted to go a little bit further, and I'm going to show you data now that really convinced me that I think that our model is correct, and I hope that you're convinced after this. So we wanted to take an even more reductionist approach. So here we use classical, like 1954 data from Tanzer et al. We set up creatine kinase activity assays, where you can take a lysate, any biological lysate. If there's creatine kinase activity, you can do this. You set it up with an auxiliary system, pyruvate kinase, lactate dehydrogenase. It's basically an ATPase assay, a creatine-dependent ATPase assay. And you're monitoring changes in absorbance with NADH oxidation, okay? That's the readout. We thought, okay, if there's a phosphatase, if we add phosphocreatine, the phosphatase should convert that to creatine and then kick off the reaction, okay? But first I'm just going to show you the CKB data. Okay, so that's the schematic. We use the same approach, the same kind of rescue experiment paradigm, and now we're adding in blue creatine and in red phosphocreatine. And you could see basically phosphocreatine stimulates NADH oxidation just as well as creatine. So that suggests that there is some sort of phosphatase activity in these lysates. Again, in the knockouts with GFP, you get no activity. Wild type restores it. The delta MTS is gone. The catalytic debt is gone. Of course, you can maybe say, well, maybe your powder is contaminated with creatine. That's probably not the case, because you could take cytosolic extracts and you lose all ability to get any activity from phosphocreatine. You only get it with creatine in the blue. And again, the wild type and the delta mutant are basically identical. So this data really confirms, I think, pretty solidly that CKB, right, you knock it out in mitochondria, there's no creatine kinase activity. CKB is the dominant creatine kinase, at least in mirroring brown adipose tissue. Okay, what about TNAP? Bruce identified TNAP. We thought, okay, we can use this now, this system. So we generated knockdowns of TNAP, 2 independent hairpins. We get nice depletion of ALPL, which is the gene encoding TNAP. CKB mRNA levels are unchanged. Here we're looking at protein levels inside protease-protected mitochondria. You can see TNAP is there. These are the 2 independent knockdowns, so we get nice depletion in mitochondria. Here, so we validate our antibodies in the lab, so we know that this is really TNAP. We also use CKB knockouts as a control to validate CKB, so we could see at the protein level CKB is unchanged. So now we do our assays in LAC-Z. You could see in blue, this is creatine. In red, this is phosphocreatine. You get similar levels of NADH oxidation. If you do knockdown of ALPL, shRNA1 or 2, the creatine-dependent rates don't change, but phosphocreatine NADH oxidation, it's specifically depleted. So this demonstrates the TNAP is probably at least the major phosphatase here in these extracts. And again, you could take cytosolic extracts, you get no activity with phosphocreatine, and knockdown of ALPL doesn't change creatine-dependent NADH oxidation. Okay, so that was a little bit of a detour, but I want to go back to a story that we're working on that's under revision, which we think is important, is to understand how the FCC is regulated under the paradigms of thermogenesis, meaning that it has to be adaptive and facultative. And what I mean by that is, cold, the most powerful stimulus of thermogenesis is sensed by the brain. Sympathetic nerves release noradrenaline, acts on brown adipose tissue. Noradrenaline will act on existing thermogenic adipocytes to regulate fuel liberation, changes in protein activity, increasing thermogenic capacity within seconds to minutes. This is fast. It's called facultative. Adaptive is transcriptional reconfiguration of existing adipocytes, right? And maybe even adipocyte precursor cells that undergo de novo differentiation into thermogenic adipocytes. These are adaptive. So how does the FCC fit within these well-established paradigms of thermogenesis? So the first thing we wanted to address was, are the genes of the creatine cycle regulated by the sympathetic nervous system? So we used a unilateral denervation approach where we unilaterally denervate one lobe of the brown fat, the other one is left intact. You could see in the sham, under cold treatment, you could see CKB is elevated, ALPL, PGC1 alpha is elevated. This will become clear why I'm going to be talking about PGC1 later on, but importantly, in the denervated lobes of the same animal, that induction is blunted. Okay, cool. So I don't have time to show, but we did a GPCR screen, because GPCRs are important for thermogenic activation. We asked the question, which GPCRs are enriched in brown fat and which ones are the most abundant? And we came out, one of the top hits was the alpha adrenergic receptor, ADRA1A. So here I'm showing you RNA sequencing data from brown adipose tissue of mice at 30 degrees versus 6 degrees cold, treated with either vehicle or an irreversible alpha adrenergic receptor inhibitor, PBZ, okay? And the cluster that I want you to focus on is this cluster here of 292 genes. These are genes that go up with cold in an alpha adrenergic receptor dependent manner. And it was really nice that CKB was the second top hit. ALPL is in this list, PGC1 alpha, ERR, okay? So this seemed to be really interesting. We then integrated this. We thought, okay, maybe we can identify the transcriptional regulators of this response. We repeated this, the same 4 groups, but now used ATAC sequencing. We looked at the differentially accessible regions of chromatin, only focusing on cluster 4 genes, and we asked, what are the motifs there, and what are the motifs, specifically, what are the motifs that are only present in both CKB and ALPL, okay? Which transcription factors coordinately regulate the genes of the fetal creatine cycle? And maybe not surprisingly, but also gratifyingly, we identify EBF2 and estrogen-related receptors, right? These are known effectors of the cold response, work from Patrick Seal and many others on the estrogen-related receptors. Okay, but no one has really tested, everyone looks at UCP1, and no one has definitely tested the requirement of these factors in vivo. So we teamed up with Patrick Seal, we got hold of fat-specific EBF1/2 double knockout mice. You could see room temperature versus cold, CKB is elevated in the cold, ALPL elevated in the cold, UCP1 in the cold, and these effects are completely blocked in an EBF1/2 fat-specific double knockout mouse. What about the estrogen-related receptors? So we teamed up with Nastasia Crowley and Vincent Giguère. We made double knockouts. We have to make double knockouts of these because they can compensate from one another. That's why. So fat-specific, again, CKB is upregulated in the cold. It's completely blocked. A little bit of an interesting wrinkle here that ALPL is actually hyper-induced in the ERR alpha gamma double knockout. And I'm not showing you this, but PGC1 alpha is also hyper-induced in the ERR alpha gamma. Okay. In our RNA-seq data, we saw PGC and the fetal creatine cycle genes, so we thought, well, maybe PGC is coordinating everything here. So we imported PGC1 alpha flox mice from genus Stahl. We crossed them to an inducible UCP1 cream model. And here we get the same level of diminution of PGC as we do with phenoxybenzamine treatment with RNA-seq. And now CKB is increased, it's decreased with PGC1 knockout, ALPL as well, decreased in the knockout, and UCP1 is actually hyper-induced. So PGC1 alpha, I think everyone thinks that it's important for UCP1, but it's obviously, PGC is dispensable. I mean, there's obviously workarounds to increase UCP1. Okay, so at the protein level, we could see this as well in the PGC knockout, CKB. It's very easy for us to detect increases in CKB. here in the cold. In the PGC knockouts, it's largely abolished. TNAP as well, nicely increased, gone. UCP, it's increased, but it's not really different between the wild type and knockouts. Okay, so this is the model that we're working with right now. We think cold is activating not beta, only beta adrenergic receptors. We think it's also acting through alpha adrenergic receptors. Somehow, this is increasing PGC1 alpha mRNA abundance. Maybe there's translational control. We're really interested in this arrow, but we have no idea what's going on. PGC then co-activates ERRs for CKB regulation, and we think PGC1 alpha co-regulates an as of yet undetermined transcription factor for ALPL, and of course all of this is under the control of EBF, which allows these factors to bind. Okay. So then we were thinking, okay, what about facultative? Noradrenaline regulates both adaptive, long-term effects on thermogenesis, but also the quick thermogenesis. So maybe the alpha adrenergic receptor is also responsible for controlling thermogenesis through the FCC. Okay, and what I want to show you is a couple slides. So this is a case report of an individual with a pheochromocytoma. This is a catecholamine-secreting tumor, and you could see massive browning of the white fat, brown adipose tissue. This is looking at glucose uptake. These patients have to undergo alpha adrenergic receptor blockade. They actually use phenoxybenzamine, the same compound that we've been using, prior to resection of the tumors. And you could see 2 weeks of alpha adrenergic receptor blockade, the glucose uptake in the fat is largely abolished. This to me says two things. The alpha adrenergic receptor is probably important in human adipose tissue metabolism, and this also tells me the capacity that human adipose tissue has to maximize or increase its metabolism in adipose tissue, right? So the other work I want to highlight here is from Barbara Cannon and Jan Nattergaard, right? So we're not working in a vacuum here. Lots of other groups have implicated the alpha adrenergic receptor, but the effector pathway has never been identified, right? So we're kind of taking on from their work. And this is literally where the field has stopped in 1997. But you'll see this sort of data later on. So this is acutely isolated brown adipocytes from a mouse. Treat with norepinephrine, you get a nice increase in respiration. Cerazolin here, which is an alpha-1 receptor agonist, does nothing alone. If you try to mimic beta adrenergic receptor signaling with forskolin to increase cyclic AMP production, you get some thermogenesis, but it doesn't match norepinephrine. But now if you combine this with an alpha adrenergic receptor agonist, now you start to approximate the thermogenesis that you get with norepinephrine. Okay. So no one has ever identified the physical coupling of the alpha adrenergic receptor, what G protein. So we use the Brett-based approach here, where we tag ADRA1A with nanoluciferase. We use these mini G proteins so we could see, and you could see basically it's primarily coupled to G alpha Q. You might predict this based on textbooks, but no one has actually ever shown this with experiments, and that's what we're doing here. So this is ligand dependent, so vehicle is here, nothing happens, but noradrenaline, cerazolin, the alpha-1 agonist, and this new agonist I'm going to introduce now, this is an alpha-1A selective agonist. All of these induce physical coupling of ADRA1A with G-alpha-Q. We've also shown now, I'm not showing you functional coupling. If you inhibit G-alpha-Q, you block the downstream effects. So we thought this was enough evidence for us that we can make a new mouse model. And what this mouse model is, it's a fat-selective expression of a modified muscarinic receptor. Okay, this is a chemogenetic approach to activate GQ only in fat. You can monitor this with HM3DQ or M-citrine, right? This is in brown fat, subcutaneous, and perigonadal fat. You could see expression, nothing in the liver or kidney. And we thought maybe we could treat mice with a beta-3 agonist to activate the GS pathway, and DCZ, which is otherwise chemically inert, to activate G-alpha-Q only in fat. And what would be the consequence? So this is the data. So this is an acute injection, one single injection. CL is in gray. Combination of CL plus DCZ to activate both G-alpha S and Q. You get a much greater increase of sustained energy expenditure. And importantly, we cross these to conditional CKB conditional CKB mice, and when you knock out CKB, this effect is largely abolished. Then we had a reviewer, which I'm very happy that they asked us to do this experiment. They asked us to ask the effect, look, what are the effects more long-term? And this is, to me, is very exciting. So here we have mice in a metabolic cage for about a week. We inject with saline. You see this is just a stress response. This is not sustained. You inject the drugs, either CL in gray or CL plus DCZ in red. This small increase here, that's what I just showed you, okay? It's sustained. But now look, over daily injections, if you look even just at the resting energy expenditure, 24 hours after, you start to see this adaptive deviation, increasing energy expenditure, total energy expenditure is increasing in an adaptive manner. And importantly, beta-3 agonism is not adaptive at all. You only get adaptive thermogenesis here in vivo with G-alpha Q and S combination. Makes sense because norepinephrine is the physiological activator of thermogenesis, and it actually binds to alpha adrenergic receptors with higher affinity than beta adrenergic receptors. We can quantify them, the total energy expenditure here. We can plot it as an ANCOVA, and you can see that at any given body weight, the energy expenditure is higher. And these effects are gone in a CKB knockout, if you knock out CKB. Okay. We want, of course, the DREAD system is expressed in white adipocytes, and maybe this is somehow regulating fuel liberation from white adipocytes into brown. We wanted to see, is this really cell intrinsic into brown adipocytes? So we adopted, we thought that this model is really important. Acutely isolated brown adipocytes. It's the most physiologically relevant system that you can use to study thermogenesis. You directly isolate adipocytes from the animal. So this is basically inspired by Barbara Cannon and Jan Nedergaard. We've done cerazlin like them, but we've also used A61603. I'm only going to show you the most selective ones. Cerazlin basically has the same effect. We isolate brown adipocytes in a Clark-type electrode. We get very nice noradrenaline-dependent respiration. A61603, right, alpha-1A agonism, nothing happens. Forskolin, you get a little bit of respiration, but then the A61603 on top of forskolin, you boost this almost to the level of noradrenaline. And importantly, in a CKB knockout, this effect is largely gone. It's about 80% depleted. With cerazlin, it's about 90% depleted. So we can say with pretty high confidence, at least in this system, that 80 to 90% of the alpha adrenergic receptor signaling cascade is mediated through CKB. Importantly, this also is mediated through TNAP. So we can do the same experiments with noradrenaline, and we can use a specific TNAP inhibitor. We can blunt noradrenaline-dependent respiration by blocking TNAP activity, and we also block this specifically when we combine the forskolin plus A61603. So together, of course, if it was just the CKB data, you might say, well, maybe CKB is in the cytosol and something else is going on. But the fact that this also happens with TNAP, I think the most straightforward answer is that it's probably the FCC. Alpha Q, G-alpha Q signaling pathway signals through IP3. IP3 engages IP3 receptors on the endoplasmic reticulum to release calcium. And we've identified the G-alpha Q pathway downstream of ADRO1A, so we thought, well, maybe calcium has some sort of effect on the FCC. And older literature, the older TNAP literature actually showed with different substrates, shows that calcium can actually activate TNAP, but no one's obviously ever tried with phosphocreatine. So we did this experiment with physiological levels of calcium chloride that are found in the microenvironment at the MAMS, ER mitochondrial contact sites, and essentially calcium doubles the efficiency of TNAP primarily by increasing the affinity, decreases the Km primarily. Much more work to be done, but this links, I think, gives us some toe in the door of how ADRO1A signaling mechanistically links to the fetal creatine cycle. So this is conclusions and working model. I've kind of gone through the transcriptional model. We think cold is activating both beta and alpha adrenergic receptors through a mechanism that we're really interested in, but we don't understand. This impinges somehow on PGC1 abundance that co-activates ERR to regulate CKB. PGC1 co-activates an as of yet undetermined transcription factor for ALPL induction. EBF, of course, controls all of this. And then on the facultative side, cold activates beta and alpha adrenergic receptors. GS and GQ signaling, respectively, is induced. You get cyclic KMP, lipolysis. Fatty acids obviously activate UCP1. There's a story, it's soon, not ready yet, but we think we've identified a lipolytic activator of the FCC, but here what I'm showing you is that calcium has probably an entry point into the FCC. So we think this is a way to rationalize how norepinephrine, the physiological activator of thermogenesis, activates multiple pathways of thermogenesis, and that's what I want to close with, maybe some implications for the field to think about. I think that it's important for us as a field to think about governing mechanisms to promote all pathways of thermogenesis, not just UCP1, not just alternative. This should be really a primary goal of the field. And I think this new chemogenetic model, I hope that it really takes on and people import it into their lab, because I think obviously triggering the orchestration of GQ and GS, that seems to be adaptive. GS alone is not adaptive, and I think we might identify new thermogenic factors by using this model, or reveal ones that were missed because we were simply only looking at the beta-3 pathway. So I'll finish. Most of the work I presented here was from a very talented postdoc in my lab, Janan Rabbani. The reductionist approaches was with Faz Hussein, a PhD student in my lab. This is my lab now. Grateful to all the collaborators and funders. Anyone interested in this type of work, PhD, postdoc, please feel free to come talk to me. I'm here for the whole meeting, and I'd love to take questions. Thanks.
That was very exciting, Lawrence. So I want to kind of ask you a question from 30,000 feet. There was great excitement about beta-3 adrenergic agonists, possibly even in humans in the early 2000s, and the results were largely disappointing, although Aaron Sipos has been revisiting some of that recently. I guess in light of that, what are the prospects for activating and increasing the amount of thermogenic fat through an alpha adrenergic pathway with or without adding beta? And can you enlighten us as to the human pharmacology of alpha adrenergic agonism?
Pharmacology of alpha.
Well, in other words, what happens if you give a human being an alpha adrenergic agonist? Does their head fall off as their brown fat gets activated?
Yeah, I think the primary effects would be on the vasculature system. I think there would be massive vasoconstriction.
Is it constriction? I thought beta was the constriction.
No, alpha adrenergic receptor agonism will constrict.
Okay.
So yeah, I don't think that this is the way for therapy through an adrenergic system. But I think obviously we've spoken about this, like the mediators downstream, the IP3 mechanism, or maybe some PTMs that are regulated by these systems might be a way forward. But I think
alpha itself is not.
No, I don't. No. That's not the point here. Is just to show that this pathway is important. But yeah, time will tell. I don't know if I'm the right person to do those studies, but. I don't think it's going to be an adrenergic agonist. Yeah. I think even mirabragon has a lot of off-target effects.
Oh yeah.
Yeah.
Yeah. I mean, the trouble is beta receptors and alpha receptors are present.
Yeah, exactly.
They're super pleiotropic. But
yeah.
Lawrence, along the same lines, did you try this in an animal, in a mouse, trying to combine these two? Because I saw that you did it in isolated cells, right? So
yeah,
did you try in vivo or no? Or does that work?
That's the chemogenetic approach. That's the only way to do it in a fat selective manner. We tried injecting phenylephrine. The mice didn't like that. They overheated and died. So phenoxybenzamine, some of the mice die as well, but that's because they vasodilate and their heat is dissipated. So you can't control. I mean, those are really dirty experiments. But the chemogenetic approach is fat selective. And we've done all the controls there, right? We can take Cre-negative mice or DREAD-negative mice and inject DCZ, and it has no effect at all. So that's the best approach to model alpha, or I shouldn't say alpha. It's modeling GQ. I mean, there could be other receptors that are coupled to GQ that might do this. It might do this even better. But in terms of, I think, the physiological adaptation to cold, noradrenaline is acting primarily through alpha and beta.
Okay. So I think time is good. Thank you very much, Lawrence.
Okay, our next speaker is Dr. David Wright. So David did his master's in exercise physiology at Arizona State University. And then he went on to do a PhD in human bioenergetics at Ball State University. And then he did a postdoc at Washington University School of Medicine in St. Louis under the supervision of Dr. John Halazzi. So he's currently a professor at the Department of Human Health and Nutritional Sciences at the University of Guelph. And I just learned, talking to him, that he is going to UBC, right? So David, thank you for accepting this, and we look forward to your presentation.
Great.
Take it away.
Great. Thanks, Rolando. Thanks to Rolando for the very nice introduction, and thanks to the organizers for giving me an opportunity to present some of the work that's been going on in my lab over the last couple of years. So just a kind of brief overview of what I'm going to talk about today, exercise and the browning of white adipose tissue, and this is going to be work from both my lab and others. The necessity of inguinal white adipose tissue for the effects of exercise, and then recent work from a recently graduated PhD student in my lab, Greg Mackay, looking at beneficial metabolic effects of activating either cold or cold-activated pathways. So I think Dr. Stanford did a very nice job earlier today kind of setting the stage for a lot of what I'm going to talk about. But what are we talking about when we're talking about white adipose tissue in a rodent? And so typically, folks in the field are studying either subcutaneous depot, so this would be inguinal white adipose tissue, which is kind of found in the inside of the thigh. They'll study an abdominal adipose tissue depot. If you're looking at a male rodent, this is typically epididymal adipose tissue. And then, as Dr. Stanford touched on earlier in her talk today, interscapular brown adipose tissue. In a very oversimplified view, we kind of view white adipose tissue on a continuum. On one end of the continuum, you have white adipose tissue, which is characterized by large unilocular fat cells. And then on the other end of the spectrum, we have brown adipose tissue. This is characterized by high mitochondrial content, thermogenic capacity, multilocular fat cells, and the presence of uncoupling protein 1. And then an intermediate would be beige white adipose tissue. So this would be white adipose tissue that's interspersed with pockets of multilocular fat cells that are positive for uncoupling protein 1. Since this is an exercise meeting, I think it's important when we're talking about the effects of exercise on adipose tissue to go back to the beginning. And we've known for probably about over 50 years now that exercise, regularly performed endurance exercise, induces increases in mitochondrial content in skeletal muscle. This was first shown by John Halsey, who I was lucky enough to postdoc with between, I'm going to date myself here, 2002 and 2006 at WashU, but in his seminal paper published in the JBC in 1967, he was the first to show that regularly performed endurance exercise in a rat leads to a very nice increase in the activity and the content of mitochondrial proteins in skeletal muscle. And so you can see here that there's probably close to a doubling in cytochrome C oxidase and succinate oxidase, and this was shown in both the gastrocnemius and the soleus muscle from exercise compared to sedentary animals. If we fast forward to 1991, and we look at the effects of exercise on white adipose tissue. And this is work that Bente Stalnick did while she was working in Henrik Galbo's lab. And if you go to the introduction of their paper, they were kind of the first that I could find in the literature anyways to hint that exercise might induce a browning of white adipose tissue. So they state that during our fat cell studies, we noticed that a mitochondria-enriched fraction of the crude homogenate of white fat cells appeared more brown when prepared from trained rats. So we've known since the early 90s that perhaps exercise could be causing the browning of white adipose tissue. And in this particular study, I'm showing the data here, this is cytochrome C oxidase activity, and looking at it in abdominal adipose tissue from rats that underwent intensive swim training compared to a sedentary control. Obviously, swim training has its limitations in terms of clinical translatability to human population, but this has been a fairly consistent finding in terms of exercise being able to increase markers of mitochondrial biogenesis and browning in white adipose tissue using various models of exercise. So the group from Ohio State in 2011, using forced treadmill exercise, showed that exercise training increased the expression of UCP1 in white adipose tissue. My lab, Dr. Goodyear's lab, and Dr. Spiegelman's lab using voluntary wheel running have shown similar effects of regularly performed exercise in the browning of white adipose tissue. And I found this interesting, published a couple of years ago from a group in Brazil, showed that a model of resistance training also caused similar adaptations in white adipose tissue. And so a lot of these studies have hinted at an association between the beneficial metabolic effects of exercise and the browning of white adipose tissue, but haven't been able to show causality, whether or not these adaptations in adipose tissue with regularly performed exercise, whether it's resistance training or aerobic training, is driving the beneficial metabolic effects. And so this is work that Dr. Stanford did when she was a postdoc in Dr. Goodyear's lab, and I think this is a very nice study showing that adaptations in white adipose tissue with regularly performed exercise are sufficient to induce a beneficial metabolic health outcome. So what they did is they gave mice access to a voluntary running wheel for a couple of weeks, and as you can see here, they see a nice increase in the expression of UCP1 in inguinal white adipose tissue. They then took the adipose tissue from the trained animal, subcutaneous adipose tissue from the trained animal, and transplanted it into the abdominal cavity of recipient mice. So these recipient mice were inactive or sedentary, and then they tracked these animals for 9, 14, and 28 days and measured changes in glucose tolerance. And remarkably, what they're able to demonstrate is that in the mice receiving the fat transplant from the animals that had undergone voluntary wheel running previously, that there's fairly marked improvements, at least over the first 2 weeks post-transplant, in glucose tolerance. So this really, I think, is the first bit of evidence to suggest that regularly performed exercise is perhaps sufficient to induce some metabolic benefit. And so I had a PhD student in my lab, Will Pepler, at the time, and we were interested in this paper, and he wanted to ask a related question. Based on this data, he asked the question, are changes in inguinal white adipose tissue with regularly performed exercise, are they required for the beneficial effects of exercise? So it's a sufficiency versus requirement question. And so the first thing that Will wanted to do was see if this voluntary wheel running protocol that Dr. Stanford had used in our hands was sufficient to induce the browning of white adipose tissue, and it was. So we see nice increases in UCP1 gene expression, PGC1 alpha, Ceddia. We see increases at the protein level as well in terms of increases in UCP1, PGC1, and COX-4 protein content. Qualitatively, you can see that the inguinal white adipose tissue from the voluntary wheel run animals appeared a little bit browner than the sedentary control, and you can see some qualitative differences in terms of the appearance of some multilocular fat cells in the inguinal white adipose tissue depot. All right, so we're able to confirm the work from the Goodyear lab, and so what we did next is we collaborated with Dr. Michelle Foster at Colorado State University, and we used a lipectomy model. So what we were thinking is that if adaptations to inguinal white adipose tissue with exercise were mediating the beneficial effect, if we removed the inguinal white adipose tissue and it was in fact mediating the effects of exercise, then some of the beneficial effects of exercise should be attenuated. And so we did this bilateral lipectomy procedure, and you can see we let the animals recover, so they had gained significant amounts of weight post-surgery by about 16 to 18 days. We then tracked the amount of wheel running that these animals did, and you can see when we compared the sham to the lipectomized animals, no difference in voluntary wheel running performance, and the ability of exercise to cause weight loss was completely intact in the sham versus the lipectomized animal. We then went in and we looked at indices of glucose homeostasis, whether that's ad-lib blood glucose levels in the fed state, glucose tolerance, or a short-term insulin tolerance test, and you can see that the ability of exercise to improve glucose homeostasis, at least with these endpoints that we looked at, so fed blood glucose, GTT, and ITT, was completely intact in the lipectomized animals. Similarly, when we put these animals in metabolic caging and measured respiratory exchange ratio in both the dark and the light cycle, you can see that the slight increase that we're seeing with voluntary wheel running was completely maintained in the lipectomized animals, as was the ability of exercise to increase the protein content of GLUT4, which is the primary glucose transporter isoform in skeletal muscle, and hexokinase 2, which is an important enzyme involved in glycolysis. So kind of summarizing some of this data, I think it's very clear that regularly performed exercise can increase mitochondrial content in white adipose tissue. It can induce browning, and I should say these studies have all been done at room temperature, and that will be an important point that I'll bring up in the next couple of slides. So it can induce browning. It can cause a fairly pronounced change in the adipokine profile, so hormones that adipose tissue are kicking out. But we don't think that these adaptations are necessary for the whole body systemic effects of exercise. I think the rodent data, at least with aerobic exercise training, is very consistent in terms of showing these adaptations, but the human data is a lot less convincing. I show 3 slides here. I could probably show another 10 that all more or less show the same thing. And so if you exercise train human participants and look at changes or indices of mitochondrial biogenesis or browning in subcutaneous adipose tissue from a human, it's all pretty much been null results. So you can't really recapitulate in human what we see in a rodent, which really begs the question, what explains these differences? Obviously, a mouse or a rat isn't a human, and there are some pretty marked differences in gene expression profiles in adipose tissue from a mouse versus a human. And so this is work that was published about 5 to 10 years ago from Ken Walsh's group, and they're looking at gene expression profiles in adipose tissue from a mouse that was fed a high-fat diet and in humans with obesity. So if we look at the mouse data first, just focusing on UCP1, if you look at the expression of UCP1, it's much higher in inguinal white adipose tissue, so subcutaneous depot in mouse versus epididymal white adipose tissue, which is an abdominal depot. In humans, the gene expression profile is flipped. So in humans, if you look at the expression of UCP1, it is much lower in subcutaneous adipose tissue versus omental or abdominal adipose tissue. And this becomes problematic when we're trying to, or attempting to design exercise intervention studies in humans, because abdominal adipose tissue biopsies, while you can procure them from a subcutaneous depot, ethically, it would be extremely difficult, if not impossible, to do similar experiments doing a pre-post biopsy from an omental or abdominal adipose tissue depot. So this could be one potential explanation for the discrepant findings between rodents and humans. Another could be related to how we house our animals. So animals, mice specifically, are happiest at thermoneutrality. So the thermoneutral zone is referring to the temperature at which mice don't have to engage additional processes to maintain core body temperature. Depending upon who you read, that falls somewhere in the neighborhood of about 29 to 30 degrees Celsius. The majority of animal vivaria are housed at room temperature. So this is thermoneutrality for a human. The staff and the trainees working in the animal facilities are comfortable at this temperature. However, it places a fairly significant degree of thermal stress on our mice, and this can have a pretty marked effect on mouse physiology. And this is some work from my lab. And so if you just look at total energy expenditure, this is in the light cycle and this is in the dark cycle. This is from mice that have been fed a high-fat diet for about 5 or 6 weeks. And you just look at the differences in energy expenditure and metabolic rate. It's almost double in a mouse housed at room temperature versus thermoneutrality. Similarly, this is data from a chow-fed mice, and looking at some of the neuroendocrine responses. You can see at room temperature, if we're looking at serum corticosterone levels, serum norepinephrine levels, or serum epinephrine levels, these are all essentially doubled in mice housed at room temp versus thermoneutrality. So obviously, these profound effects in mouse physiology could be explaining some of the discrepant findings that we're seeing between the mouse literature, especially in mice housed at room temperature, versus what we see in a human. And we tested this, and 3 papers all about the same time. Stefan Rahn at University of Copenhagen, Greg Mackay from my lab, and Peter Aldis, who was working with Michael Simons at the time. We all found essentially the same results, is that when you exercise an animal at thermoneutrality, the ability of exercise to induce browning is essentially absent. So this is, we think, probably explains the discrepant findings between the rodent literature when mice are housed at room temperature in comparison to the human data. And so, given this profound effect, we think, of cold on systemic and adipose tissue metabolism, we've become interested in looking at ways that perhaps we can use cold as a means to combat obesity and or insulin resistance. And several labs prior to this paper have used what they've termed ice treatment or intermittent cold exposure, where you have an animal exposed to various periods of cold. Some of the previous studies that have used this ice-based approach have used fairly long duration cold exposure, so 4 to 8 hours on average, and have housed their control animals at room temp, not thermoneutrality. So Greg Mackay in my lab designed this study. And what Greg did is we took, this is male C57 black 6J mice, and fed these mice either a low-fat or a high-fat diet for 5 weeks. And then I should say that these animals were all housed at thermoneutrality. After this initial 5-week period, they then underwent their ice treatment. So ice treatment was 1 hour per day at 4 degrees C, 5 days per week for 5 weeks. And then he measured changes in body weight and glucose homeostasis. And interestingly, what we found, or what Greg found, was that when you house mice at thermoneutrality and then you do this ice intervention, we actually see a greater weight gain with this intermittent cold exposure. And this tracked pretty nicely with increases in food intake. So these animals, when they're exposed to cold, are becoming hyperphagic, which we thought was very interesting. So as a next step to determine whether or not this increase in energy intake was driving the increase in body mass gain with ice, we did a pair feeding experiment. So we repeated the high-fat diet intervention and then exposed mice using our ice intervention with ad lib access to food, or we restricted food in the ice mice to the amount of the high-fat diet control. And you can see that by limiting the amount of food that the ice pair-fed mice were given, we were completely able to prevent ice-induced increases in body mass, suggesting that this is driven by hyperphagia. We also assessed indices or markers of whole body glucose homeostasis. We found slight but significant increases in glucose tolerance in the mice that had undergone ice treatment. What was interesting, though, is that this occurred independent of any changes in insulin tolerance or whole body insulin action, which suggested to us that there could be an effect of ice in driving insulin secretion. And so Greg went back and he did an acute ice experiment. So these are in naive animals, low-fat diet or high-fat diet for 5 weeks, a single exposure to the cold, and then killed immediately post and analyzed serum. You can see that there's a large increase in corticosterone, and interestingly, there's a main effect of ice to reduce insulin secretion. So what we were thinking was that perhaps with each bout of ice, this was providing the pancreas a period of rest, and that when the animals were then subsequently challenged with the glucose challenge, we'd see a larger increase in insulin response to that glucose challenge, and this is exactly what Greg found. So we repeated the experiments again, and went ahead, challenged the animals with glucose, and took serum 30 minutes post-glucose challenge, and you can see that there's a main effect of ice to increase insulin. This increase in insulin could be due to potentially one of two things. One, increases in insulin secretion and or increases in insulin clearance or degradation. So he also measured C-peptide, and it seemed to follow the similar pattern. So what Greg concluded was that this is probably increases in insulin secretion that are mediating this increase in serum insulin. And so if we go back to this, what Greg was able to show was that increases in weight gain occurred secondary to increases in energy intake, and that we saw improvements in glucose tolerance in parallel with increases in the insulin response to a glucose challenge. And so based on this, Greg's next question, and I think it was a wise one, was can we mimic the effects of cold without increasing food intake? And so he became interested in studying a compound called menthol. Menthol is an agonist of a voltage-gated cation channel and temperature sensor called TRMP8. And previous studies have used this compound and have shown increases in energy expenditure. However, they've either injected the compound via IP or sub-Q injections directly into the animal, or they've incorporated menthol into the diet. So Greg was interested in using an approach that would kind of mimic the way that the animal would actually experience cold. So Greg took mice and used topical menthol applications. So applying menthol to the back of the animal and on top of the inguinal white adipose tissue fat depot. And he did this in both TRPM8 wild type and knockout animals. And you can see that with an acute exposure to menthol, so this is within about 30 minutes post-topical administration, there's a pretty nice increase in VO2. There's an increase in heat production. This is completely absent in the TRPM8 knockout animals, suggesting that the effects of menthol that we're seeing are mediated by TRMP8. He also repeated this in whole body UCP1 knockout mice. I should say that all these experiments were completed at thermoneutrality as well. And in the UCP1 knockout mice, in the wild type, we see an increase in VO2 and in heat production that was absent in the UCP1 knockouts. Now, a caveat to our UCP1 knockout data, and this is work from Dr. Kazak and Dr. Spiegelman, is that the whole body UCP1 knockout, especially if you study them under cold stress, if you look at mitochondrial respiratory chain proteins in brown adipose tissue, they're markedly reduced. So there was a chance that what we were seeing might not be UCP1 dependent at all, but could be secondary to a degree of mitochondrial dysfunction in the whole body UCP1 knockout. So we kind of wanted to check that. This is unpublished data from a PhD student in my lab, Kyle Medak. And I think our data is pretty consistent with what Dr. Kazak found. So we had wild type mice, this is in the white bar. We had UCP1 knockout animals in the purple bar. And we treated them with the beta-3 adrenergic agonist once per day for 7 days, CL316243 or CL. And you can see that under CL-stimulated conditions, we do see pretty marked reductions in markers of mitochondrial content in brown adipose tissue. However, under the saline-treated conditions, we don't. So given that, we think that the absence of menthol to increase energy expenditure in the UCP1 knockout animal is most likely due to an absence of UCP1 and perhaps not due to a more generalized mitochondrial dysfunction. Though I should say these are pretty limited readouts that we looked at. And so the next question was, are these effects of menthol, are they direct or are they indirect? Previous work from a couple of groups using 3T3 adipocytes has shown that if you take a 3T3 adipocyte and dump menthol directly on those fat cells, you can see increases in lipolysis. So we did somewhat similar experiments. So we took explants of either brown adipose tissue, inguinal white adipose tissue, or epididymal white adipose tissue, and then measured the accumulation of either non-esterified fatty acid or glycerol in the culture media. And we treated these explants with either vehicle control, low-dose or high-dose menthol, and we used beta-3 adrenergic agonist, CL, as a positive control. Clearly, our culture conditions were working because we see a very nice CL effect in all 3 tissue depots. However, there's no direct effect, at least at the time points that we were looking at, in any of these 3 tissues. So this led Greg to hypothesize that the effects of menthol treatment in vivo are most likely secondary to an increase in sympathetic outflow. And so to test this, what he did is he pre-treated animals with a compound called 6-OHDA. 6-OHDA destroys, at least when delivered peripherally, destroys norepinephrine producing neurons. And when you pre-treat an animal with 6-OHDA, you can see that the ability of menthol to increase VO2 and heat production is somewhat abrogated. And as our positive control, you can clearly see that 6-OHDA completely prevents the ability of menthol to increase serum norepinephrine levels and norepinephrine accumulation in brown adipose tissue as well. And I think, most importantly, these were all acute experiments we did. What we wanted to determine next were these repeated transient increases in energy expenditure with menthol, was it sufficient to protect against weight gain? These are in chow-fed animals, chow-fed mice. We've also shown the same thing under high-fat diet conditions, and you can see that with repeated menthol treatment, and he went once per day for 14 days, you can see that total weight gain was attenuated by about 50%. And importantly, this occurred independent of any changes in food intake. So we think that what we're seeing here is due to an effect on increases in energy expenditure. And so this is our working model, that if you take an animal and you treat topically with menthol, that it's signaling through a TRPM8 dependent mechanism, signals centrally to increase norepinephrine secretion, which is then driving increases in energy expenditure, perhaps through a UCP1 brown adipose tissue dependent pathway. Now, if we're to translate this to humans, I think there are some caveats that we need to take into consideration. First and foremost among these, especially under conditions of obesity and pre-existing insulin resistance, is that when you treat an animal with menthol, you see not huge increases, but still statistically significant increases in blood glucose. And this is probably due to increases in hepatic glucose production. So if this is an approach to treat insulin resistance, then I think this is something that's going to have to be overcome. And then the most important slide for last is the good folks in the lab that actually did the work. Will Pepler, Greg Mackay, Kyle Madak, Carly Newth, Hisham Shamshun, Logan Townsend, who's in the audience, collaborators Christine Doucette, Michelle Foster, and Jabranko Carr, and then our funders. And I think I should have a few minutes for questions. Thank you.
David, that was really nice. I just had, John Speakman has challenged Barbara and Cannon on Netagards in terms of like optimal housing temperature. I think he suggests if there's bedding or nesting, that actually 22 to 24 degrees is sufficient. Yeah. 30 degrees might be too hot. So I'm just wondering what your thoughts are on that.
That it is contentious for sure. And so all of our studies that I've shown, mice were individually housed. I don't know if that's a consideration. I think as long as you're closer to 28, 29, it's probably better than being closer to 20, 21, 22, even with group housing, I think.
Yeah, thank you very much for a fascinating talk. I have the question, okay, in the mice, the temperature plays a role in terms of UCP1 expression, but what about the human experiments? If we make humans exercise in the cold or in the heat, would you hypothesize that there are some temperatures where you actually see the browning effect in response to exercise? Do we do these experiments right? And could that be a reason, or?
Yeah, potentially. So I think it's probably, or it could be related to a depot-specific issue that I mentioned before. So if we could somehow get to omental or abdominal adipose tissue depots in the human pre-post, that could answer that question. We have some inconclusive data anyways, where we took mice at thermoneutrality and then treated them with menthol and then exercised them. So inconclusive at this point whether or not kind of mimicking that cold environment then induced browning of white adipose tissue in a rodent model where you don't see browning. We need to repeat those
Okay, thank
experiments
you.
then. Yep, you're welcome.
Hi, I'm Lykke Sylow from the University of Copenhagen.
Yep.
Nice talk. We share an interest in trying to mimic the human condition a little better in mice by thermoneutral housing.
Yep.
And something I've thought a lot about, and we have not tested it, so I'm just curious if you have, is if you know how changing the temperature for the mouse affects diurnal rhythm of the mouse, so
Oof.
feeding behavior during the day or maybe even exercise during the night.
And so the exercise, the temperature-dependent effects on exercise are really interesting because I know your paper and our paper don't line up. So we found that when you house a mouse at thermoneutrality versus room temperature, they run more. And your group found the opposite, right? And so there's a male-female comparison there as well. I'm not sure. I for sure there's something going on, and I think it needs to be really kind of thoroughly teased out because, I mean, housing temperature is going to affect probably physical activity one way or the other. It's clearly impacting food intake, and it's clearly impacting energy expenditure, right?
Yeah. So mice, they run, like, they go on and they all go off. And
Yeah.
so have you checked if that rhythm has changed?
Our system that we use is pretty archaic, so we've ordered new wheels, and we should be able to tease that out more specifically. Yeah,
Well, thank you.
thanks.
Thank you, David.
Excuse me, okay. Okay, I'm going to close the session with a discussion about what happens in the white adipose tissue with browning. If it's really thermogenic or what I think it's more like a metabolic remodeling. So I'm going to present some data that contrasts both views, and I hope to convince you that what happens in the adipose tissue is not really, I mean, the browning has a lot more with alterations in metabolism than in the thermogenic response of the tissue. So plasticity in the adipose tissue has for a long time been recognized because there are many studies, and I think the previous presenters mentioned that several times. But I always like to illustrate this with a study done by Dr. Saverio Cinti in Italy, where he actually put mice under two conditions, at 20 degrees and at 4 degrees Celsius. And he dissected the animals, and then he actually put the adipose, the fat depots, in their respective places. And it's obvious that under cold exposure, there was a very obvious
alteration in the color of the white adipose. Not only the brown adipose tissue actually responded to it, but also the white. And this brought up the idea that, well, if the white adipose tissue can be altered, if you can switch the metabolic response of the tissue and turn it more into a brown like fat depot, you might be able to dissipate energy as opposed to store energy, which is the classical view of the white adipose tissue. So there were several other studies, and also Dr. Wright also mentioned before, but this is just one of the studies that showed that exercise actually could increase browning or this alteration in white adipose tissue. Not only the cold exposure, but it could also be induced by exercise. And in this study, they also measured mitochondrial number and mitochondrial volume. And there was a nice increase in mitochondrial content in these tissues with exercise. And with immunohistochemistry, it was clear that there were some UCP positive cells in the tissue. So exercise was clearly a means to induce this alteration. So with this in mind, what we did is like, what if we actually use a very controlled system to exercise animals and see if we can induce a similar response? And we used the rat as a model here because we could control and we could get them to exercise quite well and control the exercise intensity, volume, and duration. And these rats, we selected them in different groups where we gave them either standard chow or high-fat diets, and they were either sedentary or exercised. And these animals, we could get them to exercise regularly 5 days a week, 1 hour per day at approximately 75% of VO2 max. And these rats were running easily at some point at 40 meters per minute with 10% inclination. So they were really exercising. It was not a mild type of, it was a moderate and high intensity exercise with these animals. And they tolerated it very well. So at the end of the study, what we did is we isolated the fat depots. And one thing that really stood out when we did these experiments was that the subcutaneous inguinal fat, which we extracted and we put it here just for visual, and we noticed that there was a clear change in color of the tissue, especially in the middle portion of the subcutaneous inguinal fat. The extremities of the tissue didn't change much in color, but it was more in the middle portion of the tissue, as indicated here. You can see that it becomes quite darker and more like a brown-like kind of tissue. And so what we also looked in these animals was the brown adipose tissue. So we knew that apparently there was, or the visual indicated that there was a browning effect in the subcutaneous. But we also wanted to look at the interscapular brown adipose tissue. And we also excised, we extracted, and then looked at, when we dissected the tissue, we saw that there was the sedentary and the exercise, they were significant. The exercise animals had a much smaller brown adipose tissue. The high fat, there is an increase in the size. It was obvious also, but when we exercised the animals, there was a significant reduction in the size. So then we select, well, the size cannot really tell us much because it may not really represent what's going on in the cell. So we went on and we zoomed in the cells and we looked at the structure of the cells, and we saw that with exercise, surprisingly to us, what happened was there was an invasion of these white-like fat cells with unilocular adipocytes as opposed to this multilocular typical brown adipose tissue. And again, with high fat, they had a little bit more fat, but they were not as clearly invaded with fat as we saw with exercise. And this was the same with the high fat when we exercised them. So there was something going on in the brown adipose tissue that really indicated to us that instead of becoming more thermogenic, it was, if anything, partly becoming less thermogenic, because we were actually increasing the amount of fat that is deposited within the fat depot. So when we also measured, I mean, here is a representation, a graphic representation of the weight of the tissue. So the IBEC was significantly lower in the exercised animal. And when we looked, we were able to measure the number of this and the size of this unilocular fat cells that were invading the tissue, and they were significantly higher also in the exercised animal versus the non-exercised. So this indicated to us that there was, again, instead of having an activation of brown or thermogenesis in the brown adipose tissue, it was actually doing the reverse. So we are actually accumulating more fat within the interscapular brown, which is typical of a brown fat depot. So we also ran a series of other experiments where we tested, actually, we did functional experiments with isolated adipocytes from the interscapular brown adipose tissue, and we found that there was no, not only the morphology was changing, but also the function, especially when we measured, we gave substrate to the isolated adipocytes and measured the capacity to oxidize either glucose or fatty acids, and they were significantly reduced. And also when we measured UCP1 content, it was also lower in this interscapular brown adipocytes of exercised animals. So shortly after we published that paper, this study came up in humans, and it was already brought up by Dr. Stanford here, where they actually measured in endurance-trained, long-distance cycling, swimming, running, cycling, and swimming athletes. And they had at least been training 3 times a week for the last 2 years. And they had a VO2 max of approximately higher than 55 mL per minute per kilogram. So these are pretty good, very well-trained subjects. And when they compared the rate of glucose uptake in brown adipose tissue in this subjects, they found that the trained was much lower than what you see in the untrained. So this really kind of went well or coincided with what we saw in rats. It was a similar response. And again, I think it has also a lot to do with the type of exercise, the intensity, and how we controlled the intervention with exercise, because it was, I would say, significantly effective in improving the oxygen consumption in these animals. And again, in humans, it shows a similar kind of response that we saw with rats. So I don't think there is, depending on how you manipulate or how you approach the problem, you may actually get similar results that are very similar to what we see in humans. So then we shift our focus, because let's say the brown adipose tissue, the typical brown adipose tissue may not be, may be affected in a way that it's less thermogenic. But what happens to the subcutaneous inguinal or the subcutaneous fat? Because that's another one that we saw very interesting effect with exercise. And again, when we zoomed in and we looked in those areas, in those central areas of the subcutaneous inguinal fat, so this is the sedentary animal, so this is the typical white fat with the unilocular shape, format. And then when we looked at in those areas in the middle portion, we saw that we had these islands of multilocular adipocytes. So there was a clear change in the morphology of those cells. And again, when we looked the IBEC, just for comparison here, we see that the IBEC, again, the typical sedentary animal was that multilocular shape or form, and then the invasion of white fat here. But in the subcutaneous inguinal, it was a clear change towards a more multilocular morphology. So we went on and said, look, is this also accompanied by increases in the molecular structure or proteins or the components that actually are related to thermogenesis? So we looked at PGC1 alpha in the standard chow or low-fat rat, and sedentary versus exercise. There was a marked increase in PGC1 alpha in the subcutaneous inguinal fat. And in the high-fat exercised animals also, although a much lower response compared to the low-fat animal. And then we looked at UCP1. We saw a very robust increase in UCP1, the protein itself.
And in the sedentary high fat, we couldn't barely, almost no detection of UCP1. But with exercise, we were able to see some that increased there also. So we rescued some of that UCP1. So, and then again, this is the graphic representation of the alterations in these proteins that we see up here. So everything was pointing to us to something that was increasing. If you have an increase in UCP1, we think, well, it might be because it's uncoupling the tissue and it's also producing, has a thermogenic response. So, but the problem in the thing that was really, I think the elephant in the room when we looked at this data was, if exercise is highly thermogenic in itself, so when you exercise, I think there is no question that you produce a lot of heat. So you are increasing thermogenesis by exercise itself. So why is it that you would actually increase in the subcutaneous tissue thermogenesis when you're actually trying to eliminate heat because you're producing heat with muscle contractions? So we questioned, and some other people, we're not the only ones that came up with these questions, but our idea was, are these exercise-induced adaptations in subcutaneous inguinal fat truly thermogenic? Is that really because the tissue is altering its function to become more thermogenic? Because it was kind of antagonistic with the idea of what exercise itself does. So then there's a student in my lab that she actually, Diane, what she did is we took the rats and we said, well, let's put the rats actually in a condition where we really know that they need to be thermogenic, that they need to increase the capacity to produce heat. So we took the rats and we put them in the cold. So we exposed them for 7 days to 4 degrees Celsius. So we collected blood and food intake daily for 7 days. And we did a tissue extraction and analysis at the end of this 7-day period. And again, we saw similar kind of responses that we saw with exercise because, well, at 22 degrees, we had that typical morphology. So the subcutaneous inguinal, there was no, the typical unilocular, and the epididymal fat also for comparison, there was no alteration. But in the IBEC, in the interscapular bat, we also had some invasion, not as much as with exercise, but we also saw some increase in this unilocular deposition of fat. And then in the subcutaneous inguinal, we had again those multilocular, those islands of cells that actually reproduced to some extent what we saw with exercise. So with this, we knew that we had a system, a model, that we had adipocytes that underwent browning. Now the question is, are they really thermogenic? I think that was the important question to answer. So first what we did is we looked at lipolysis in this. We isolated the cells, and then we exposed them. Either we measured lipolysis under basal or beta-adrenergic stimulation with CL. And again, we see here that under room temperature, we got a significant response as expected, but under cold, it went up, which is also expected. So in the subcutaneous inguinal fat, it was interesting because usually when we measure lipolysis in cells from subcutaneous and we compare with visceral fat or with epididymal fat in an animal, in a rat specifically, we see that the epididymal fat is a lot more lipolytic than the subcutaneous inguinal. But when we exposed these animals to cold, they became a lot more, there was a much more robust response. They became a lot more lipolytic. And so there was a more accentuated response in the subcutaneous inguinal than the epididymal. And the reason why we were interested in this is because the traditional idea is that in order for you to activate AMPK, to activate UCP1, you need to have fatty acids because interacting with the protein, it activates it. So we thought, well, lipolysis is an important measurement here to see what's going on. And when we looked at all the machinery that is involved in lipolysis, tyrosine hydroxylase, beta-3 adrenergic, ATGL, and
hormone-sensitive lipase, they all went up in epididymal and also in the subcutaneous inguinal. So again, it was indicating that there was some degree of at least converging towards something that was thermogenic. And then we looked at UCP1, and IBEC classically went up. And in the aortic brown adipose tissue, the same kind of response. And then when we looked in the subcutaneous inguinal fat, there was also an increase in UCP1 content. So everything was lining up with that. So we also did another experiment because we wanted to compare how much more UCP1 there is when you compare the classical interscapular brown with the subcutaneous inguinal under the same conditions. So under cold acclimated conditions, when we actually probed for UCP1 in both tissues, we ran in the same Western blot. We found that even after acclimation, and if there was the browning in the IBET, we had approximately 20 times more UCP1 than we had in subcutaneous inguinal. So even though there is an increase in UCP1, it's a very small amount that actually appears there. So this is important because the relevance of this for thermogenesis is critical here, because if you have a protein, it's a very small amounts, it might not be significantly or relevant from the point of view of thermogenesis. So then we ran another experiment because we need to test if there is uncoupling there. So we isolated again brown adipocytes from the interscapular brown adipose tissue and the cold and the control and cold exposed animals. And under basal, we measured palmitate oxidation. So we have the cells, we give labeled palmitate, and we looked at oxidation rates, and it's, as expected, again, quite high. We gave isoproterenol as a lipolytic agent. We expected to increase more, but there was not much, any difference, significant difference, although it increased compared to basal, which is also expected. And we used oligomycin to try to inhibit ATP synthase and force, if there is respiration, to force it to go, or the proton gradient to go through the UCP1. And again, that was a clear response, a clear increase, so showing that there is uncoupling, the uncoupling is increased in those cells. So then we did the same thing, but now using subcutaneous inguinal fat cells. And under basal conditions, there was no difference in rates of palmitate oxidation. When we put actually isoproterenol, instead of increasing, oxidation actually went down. So there was no additional effect or no stimulation with lipolysis in this tissue. So again, which was contrary to what we expected. And again, when we put in the presence of oligomycin, it went down. So which indicated to us that actually we have a more coupled mitochondria as opposed to uncoupled. So again, there was a kind of unexpected effect here, because why, if we have more UCP1, why is it that we don't have more uncoupling here? So I think when we looked at this, we thought, well, it might not be because these cells are becoming thermogenic, it's because this UCP1 does not really have a thermogenic effect in the subcutaneous inguinal or in this wide adipose tissue that underwent browning. So then we looked at the expression of proteins that are involved in lipid cycling, so especially for fatty acid uptake and also for thermogenesis to some extent. And so we looked at lipoprotein lipase, CD36, but also looked at PGC1 alpha. So PGC1 alpha was up, which is compatible with that increase in UCP1. But then what we looked at, we thought that, well, if there is more lipids entering the cell, so then what is the fate of those lipids? So we thought that there is an enzyme called glycerol kinase that actually, if it phosphorylates fatty acids, it can actually store in triglycerides. Again, it creates a futile cycle where you can actually have lipolysis, because we know the lipolysis is increased. But at the same time, we know that if you can phosphorylate glycerol, it can also re-sterify. So it creates a cycle of lipolysis re-sterification. And this is important because from a thermogenic perspective, in the subcutaneous inguinal fat, that could be very relevant. And this is considered the pathway that has the highest, or that consumes the highest amount of energy. So again, when we look at the gene expression of glycerol kinase, it was significantly elevated. It was almost 8-fold increase, almost 10-fold higher, 8-fold higher than what the control animal was. And then we looked at the protein. So again, protein was also increased in the tissue. So the next experiment we did was, well, how do we know that this is actually functioning the way we expect it to function? So we actually incubated the cells again in the presence of glycerol, and then we looked at how much of that glycerol is incorporated into triglycerides or in lipids. And in the presence in the cold exposed animals, we see a significant incorporation of glycerol into lipids. And even when we gave glucose, because usually glucose is what is the main substrate for esterification, for the formation of glycerol 3-phosphate. And again, even in the presence of glucose, we had a much higher incorporation of glycerol into lipids. And similarly to palmitate, if you give palmitate, there was a significant increase in this cold exposed, this brown or beige adipocytes, if you will. The other important thing we wanted to look at is what is it doing to actually triglycerides in the circulation. And as I mentioned before, every day of the experiment, we collected blood and we measured triglyceride levels. And in day 1, we already saw a drastic reduction in triglycerides, circulating triglycerides. Obviously, it's not only due to incorporation or cycling of this fatty acids into triglycerol and back and forth into the subcutaneous adipose tissue, but it's an indication, or it's a response that looks very consistent with the fact that the tissues are actually able to recycle and take up quite a bit of fat, as opposed to the classical thermogenesis response that we thought in the beginning. So there was a 57% reduction in triglyceride levels in these animals, and it's important to mention that when you put them in the cold, they eat 50% more. So they were hyperphagic, they were eating a lot more, and yet the amount of triglycerides was much, much less than what you see in a non-cold exposed animal. So to conclude here, so we had the model that we came, I think the model that we're working now is we have the classical brown adipose tissue or adipocyte that has UCP1, and it is multilocular, and what it does is uses fatty acids to produce heat when thermogenesis is required. And we also have the classical white adipose tissue, those cells that maintain that unilocular shape. They have very few mitochondria, and they actually export fatty acids, and this can go to muscle and even brown adipose tissue for thermogenesis. And we have the beige adipocyte, which is in between both. And I think it has the capacity to release fatty acids through lipolysis, which is what we found. And also it can recycle glycerol and use that glycerol for esterifying fatty acids again and forming triglycerides. And this is one of the reasons probably why we have multilocular formation, because now you have smaller droplets of triglycerides due to this rate of recycling that is increased in the fat cell. And this is thermogenic because this consumes quite a bit of energy for you to form fatty acyl-CoA and also to esterify, to phosphorylate glycerol, and also for glucose to some extent, because glucose is also used for glycerol 3-phosphate. So we have at least 3 different possibilities here that increase thermogenesis in the adipocyte, not because of uncoupling. Uncoupling is probably the least relevant effect in this beige adipocyte. If there is thermogenesis, from what we've seen, seems to be more
deriving from these alternative pathways or these changes, this metabolic remodeling that I mentioned before, because it changes this fat or increases the capacity of this cell to deal with lipids in a way that it releases fatty acids but also has to recycle them, because not all those fatty acids are actually being used immediately, so they have to be recycled. So what I presented here is data relating to this thermogenic response of lipid cycling. So it's a UCP1 independent mechanism of energy dissipation in brown adipose tissue happens, but it also in this white adipose tissue that became brown-like. But there are also at least two other mechanisms that could add on to this thermogenic response. One is the one that Dr. Kazak presented earlier, which is related to creatine-dependent ATP and ADP-ATP substrate cycling. So this can also be thermogenic in the cell, and again has nothing to do with the presence or absence of UCP1 in the beige adipocyte. And also another that has also come up recently, that is this SERCA2B mediated calcium cycling. So this also, through the alteration in calcium handling inside of the cell, it can also change the activity of SERCA, and this also add on or contribute to this thermogenic response. So we have different mechanisms in this beige adipocyte, if you will, or even the white adipocyte that became brown-like, that could contribute to thermogenesis, but not necessarily, again, through the classical UCP1 uncoupling mechanism that we often hear. So to conclude, so is energy dissipation through UCP1-mediated mitochondrial uncoupling thermogenically relevant in beige adipocytes? I would say that likely not, because the data does not support, at least our data does not support it. So alternatively, browning of white adipocytes appears to be a metabolic remodeling process that involves enhanced capacity to break down, export, and resynthesize triglycerides, and leading to the activation of energy-consuming pathways that can also be named futile cycles. Well, people call it futile, but they're not so futile because they have a function, they have a reason to be there. But that's the classical way of looking at these energy-consuming pathways in cells, especially in this case in this potentially beige adipocytes. So I just want to mention here the students, Daniel, who is sitting there, Shaili and Matea, they do all the work. They are really very dedicated to the lab, and Daniel and Matea, they both have posters out there. If you want to check it out, they did studies using ketogenic diet and trying to change metabolism in adipose tissue and muscle to see how that affects substrate cycling in these tissues. And obviously the funding agencies. Thank you, and if you have any questions, I'll be glad to take.
Just a question about, I might have missed it, the glycerol or the palmitate incorporation into lipids, is that sensitive to oligomycin? Can you block that with oligomycin? Because you would expect that should, if that's the consuming energy part cycle, that should be blocked by, or like triacin C?
We never tested that. That's a very good suggestion. We never really did the incorporation in the presence of oligomycin. But I don't know if it would alter the incorporation, because when we had oligomycin, it reduced the oxidation rate, but we don't know if that will affect, it may actually affect, it could even increase the cycling, because if you're not oxidizing the fatty acids that are entering the cell, they're gonna have to be diverted somewhere. So there might be more recycling. We never tested it, but that could be a good way of looking to see if there is any connection between those two pathways.
I had another question about the first slide you showed where exercise basically dramatically reduced the size of adipose tissue, and normally we consider that, oh, there's less lipid. So, but you had more lipids. So I have a couple of questions. where are the cells going? And the ones that have more lipid, are those newly developed adipocytes? I mean, do you have any idea?
We never tested for that. That could be that. I don't know if it's newly formed adipocytes. I think what we are having there is more fat being stored as opposed to being released or used. Now, when we measured fatty acid oxidation and we measured different pathways in those cells, I didn't show the data here. They were reduced. So my guess, if I were to look at that, I wouldn't say that it's new adipocytes. I would say that it's the same adipocytes that actually became more unilocular because now you don't have to recycle as much, or you don't require as much of those for the formation of for oxidation or for heat production. So that's my guess, but again, we never went that route to see if those were new adipocytes. Because actually we focused more on the subcutaneous because we wanted to see, is that browning really something that we can consider thermogenesis?
It's such fascinating work, Rolando. Have you ever thought about what would happen if you did like explant transplants between the sedentary animals and the exercise-trained animals? Like you flip the fat pads. Would that change the lipolytic rate or somehow be a disadvantage during the training adaptation? I wonder.
You mean take the fat from the trained animal and transplant to the sedentary animal and see?
And I was thinking maybe the sub-Q would be the one, because that seems to be so fascinating. Like, what are your thoughts on that? Is that technically possible?
It is possible to do.
What would you think would happen? Because I couldn't wrap my head around it.
I think that if we didn't do this, but my hypothesis would be that if you really do the transplantation after the training, I think it would be very similar to what Dr. Christine Stanford saw in mice, where she saw that it changes significantly metabolism, it improves insulin sensitivity, reduces glucose levels, and all those things. So I think that would be the case. But we did another experiment, and we never really got to continue much because we had some issues with the pandemic and everything else. What we had done a few years ago, we did lipectomy in those rats. And we used the rat, again, different, I think Dr. Wright did this in a mouse. We chose to do in a rat because if you look at the subcutaneous inguinal fat in a rat, it's a substantial amount of tissue. If you look in a mouse, it's a really strip of fat, tiny strip of fat. It's easier for you to excise and take, but I don't think it really has such a big impact, even though it undergoes browning and it does a similar alteration. But it's very small compared to the total amount of fat that the animal has. In the rat, it's different. You have a massive amount, I mean, it's not massive, but it's a pretty large amount of fat that you have in the subcutaneous inguinal. When we took that out, it made a huge difference to the animal. So we are trying to reproduce that. We are trying to repeat that and do this again. Instead, it was a different kind of question, not exactly the one that you're doing, but I would expect that it would see the same thing that was reported in mice. So, but it made a significant difference in a rat. They had lower energy expenditure. They had a whole bunch of alterations in metabolism when we took the fat out. The problem with the rat is that a lot harder for you to manage the surgery because it takes a lot more cutting and opening to be able to take all the fat out. But it's doable. We did a few experiments with that, so we need to move on to that. So, okay. Thank you very much, and have a nice evening.